Good afternoon. My name is Gina Wen. I'm a SMID-cap biotech analyst. It's my great pleasure to introduce our first speaker, Sandra Van Deventer, Chief Scientific Officer of uniQure.
Thank you very much, Gina. It's a pleasure to be here at the conference. Thank you very much for the invitation. It's also a pleasure to introduce our company to you. I will be making some forward-looking statements, as you see here, during my presentation. So, uniQure is a gene therapy company, and the mission of uniQure is to deliver curative one-time therapies that transform the lives of patients, basically long-acting therapies. So the drivers behind our strategy are, pipeline. So we have developed a pipeline of gene therapy candidates which are focused on the liver and on the CNS. We'll discuss that in a little more detail. If you want to make a product, you need to have manufacturing. That's true for any drug, but it's certainly true for gene therapy. We've been manufacturing for 20 years and 16 years in insect cell-based systems, and I think we're the leader in that respect. We have the capability to manufacture in-house all our clinical material, and we have the capacity to produce all the commercial worldwide material for our products that we're currently developing in-house. We have also invested quite a lot in enabling technologies such as promoters and other technologies that optimize gene therapy, and we have, for example, last year increased expression of proteins from liver-directed therapies by more than tenfold, just looking at promoter technologies. Our intellectual property is broad. It covers manufacturing. It also covers, for example, the PADUA mutation in the factor IX gene and many other aspects of gene therapy, and we have retained all the commercial rights on our programs worldwide. So our pipeline currently, where are we? For hemophilia B, as you probably know, we're in phase 3 with our HOPE-B study, and I will cover that in some detail. We also have a hemophilia A program that is currently in a GLP toxicology study, and we hope to file an IND for that program late this year, early next year maybe. We have disclosed our Fabry program, where we think we have a distinguished approach that we think is probably superior to ERT, definitely, and we have some other liver-directed targets. And for the CNS targets, we'll talk a little bit about Huntington's, where we are the first gene therapy company to hit clinical stage. As you know, we have cleared IND for that, and we're going to treat patients later this year. We also are rapidly developing SCA3, which is spinocerebellar ataxia number 3, which is also a large indication, actually approaching Huntington's disease and an unmet medical need patients. There's no therapy for them, and it's a very severe disease. And then other CNS-directed targets. You probably saw that very recently. We published 2 papers on ALS, and that's another program we're developing. And then we have some partnered programs with BMS. So what drives the value, we think, of the company for 2019? Well, first of all, we expect to completely enroll the global phase 3 HOPE-B trial in hemophilia B. We, as I just said, also expect to initiate dosing of patients in the phase 1/2 clinical study of AMT-130 in early Huntington's disease patients. We want to submit this IND later this year or late this year for our hemophilia A program. And we will initiate the GLP tox study for one other research program, and basically that is not a matter of thinking what we have, but it's more a matter of prioritizing what we have. So let's talk a little bit about AAV manufacturing. In Lexington, we have 80,000. We're expanding. We have 55,000 square feet. We're expanding to 80,000 square feet. We are using a 3rd-generation insect cell baculovirus manufacturing system that is widely protected by 10 patent families. We are now manufacturing in 500-liter stir tanks. We can scale up at that manufacturing facility to 2 times 2,000 liters, and as I already said, there's a strong IP on the manufacturing. So we can control this process, it's consistent all the way from preclinical. When we do mouse studies, actually the quality of the product is almost indistinguishable from what it is commercial. It's high scale, we are very cost effective and high volume, and this leads to a high quality product. AAV is the other aspect. It's our workhorse. It's not the only factor we have. Actually, we have 50 odd AAV serotypes and synthetic factors, but AAV5 is what we work with. We have been working with AAV5 now for a long time. We have long-term follow-ups demonstrating clinical safety and tolerability. We have done 4 studies with AAV5 in the past, both in the liver and in the brain. We do know that pre-existing antibodies to AAV5 do not interfere with therapeutic efficacy, so we are one of the very few gene therapy companies that doesn't select patients based on pre-existing antibodies. We basically treat everybody. We also have no T-cell responses, so we don't have to treat patients with steroids, and we've never seen T-cell responses in any patient treated with an AAV5. So that is really very favorable from an immunological point of view, but also from a market point of view, because if you exclude about 40% of your patients, such as our competitors do, of course, the implication is that your market immediately also is basically half of what it could be. So where are we with hemophilia B? So we believe that this product has the potential for a functional curative increase in factor IX activity, which we think is about 30 to 50%. We also strongly believe that the response to therapy is durable and predictable, and that there's a very low risk of immune responses, and we haven't seen any thus far in our phase 1/2 study with AMT-60 or with the phase 2b that we've done with AMT-61. And again, we have intellectual property on this product because we have 2 issued patents on Padua in the United States and 1 in Canada, and pending IP in Europe. We think we'll have the potential to be first to market. In terms of timelines, we can discuss that a little bit later. So this is the phase 2b study. Basically, we initiated that study in 3 patients to have some data and some prediction of the dose, whether our dose prediction was in the correct range. We predicted to be between 30 and 50%. We're smack in the middle of that. At 12 weeks, the mean expression was 38%, and you can see here that these curves are really very predictable in time. There's no funny ups and downs. There's no immune-mediated decline of expression, and we expect actually basically that these levels will be continuous from now on or slightly increase. And we will update mid-year the 26-week follow-up, and late in the year probably the 52-week follow-up. There were no immune responses, no patients received steroids, there were no adverse effects. All patients are off replacement therapy, and no patient has bled in that study. For the phase 3 study, the pivotal study that should lead to the BLA, that's a study including 50 patients with severe to moderate hemophilia B. We are not excluding anybody for AAV antibodies. There's a 6-month roll-in period where patients are being treated with prophy, and then we'll have the patient treated, and then basically the primary endpoint for this study is 6-month factor IX level, and the secondary endpoint here is bleed rates at 12 months. So that's basically what this study is ongoing. We reported that the first patient in this study was dosed in January, and we're pretty happy and on time with on the timelines of our enrollment, and we expect the study to be fully enrolled by the end of this year. Huntington's disease is AMT-130. Again, the target product profile, again, a one-time administration. This therapy should be really disease modifying. We are using a unique silencing platform here. It's a MyCure silencing platform that we can maybe discuss if there's any questions there, but it's unique in that it doesn't have off-target effects, and it's very strongly silencing also cells that are not directly transduced by the virus. We have shown a strong mutant Huntington protein knockdown in the deep structures. That's where the disease starts, in the striatum and in the cortex. We've shown in 5 different animal models preclinically in 4 different species that this is safe and well tolerated. We did a GLP tox study in primates using exactly the administration procedure that we're using in patients, and we've not hit any toxicity in that study. Now, we target full-length Huntington protein, as do many of our competitors, of course, but we also target the exon 1 protein, which is a small fragment that is transcribed of the exon 1 of the Huntington gene, which is a highly toxic subspecies of mutant Huntington. And in that respect, we're the only ones. There's a lot of excitement about this now because it's increasingly recognized that it's not only the mutant full-length Huntington protein, but this small fragment which is highly toxic. And again, we expect to be potentially first to market for this indication. Yeah, just to show you here, you know, the disease really starts in the putamen and in the caudate nucleus, which is basically the striatum, and spreads to the cortex. And we target those regions. Basically, I think it's very well illustrated here in the non-human primate. All the brown is the transduced, you know, the initially transduced area. And you can see that this is exactly the area where you find Huntington's disease. We also have a strong reduction of mutant Huntington in the pig, which is a much larger animal than the primate, and it has a much larger brain. We use exactly the same MRI-guided convection-enhanced delivery technology as we're doing in humans here, and we now have 12 months data of strong knockdown, not only in the areas where we want to have this knockdown, but also we show an effect on mutant Huntington knockdown in the CSF. So the IND is cleared. We are initiating the study. We'll start to dose patients first half of this year. There will be early manifest stage 1 patients, but they're enriched for CAG repeats, so we'll select patients who have more than 44 CAG repeats, and that ensures that the downward slope is predictable and fairly rapid, so you will have a higher chance of finding therapeutic efficacy. It's a multi-center randomized double-blinded study, so patients will have a sham operation and they'll have sham burr holes because we really want to blind the study in order to enable us later maybe to morph it into a study that can be used for regulatory endpoints. And the objectives now are to assess safety, tolerability, and efficacy as measured by efficacy endpoints, including clinical parameters, quantitative motor function scores, but we're also looking at MR and functional MR. We found in our preclinical models that there's a strong effect on functional MR. Actually, we restore function where it has been lost in these animal models, which I think is pretty spectacular. And then we have a range of biomarkers. And of course, patient-reported outcomes. We'll do 2 dose cohorts with a total of 25 patients. And the first dose cohort, actually, we initially were contemplating 3 dose cohorts, but we dropped the lowest dose because of all the safety and efficacy that we saw in the primate study. So basically, we stepped up a dose, and the first dose already we think will show therapeutic benefit. So what about the research pipeline here? Well, I already alluded to, we have a program in HEM-A. It's a factor 9, which has been modified so that it doesn't need factor 8 any longer. We think this is better than expressing factor 8 because factor 8 expression in the liver leads to problems that we might discuss later. And we found that early in our development program, so we have made a factor 9. It's non-factor 8 dependent. It's normally activated. It's normally inactivated, and it causes thrombin formation. And for example, if you measure it by one-stage clotting assay or thrombin generation assays, in the models we have now or in patient plasma, basically is at par with emicizumab. So we think this is really exciting because we can roll this out quickly. We don't need to change our manufacturing procedure. It's basically very much the same as it is for 61. Actually, we already have manufactured this in 500-liter scale, and it is safe in combination. We already showed that with NOVO7 and with FIBA. Remember that emicizumab in combination with FIBA is highly thrombogenic. We don't see any of that. We have a normal regulation here. And of course, this is applicable to patients with inhibitors as well. Here you can see that we've made great progress in expressing genes in the liver. In this program, for example, we use a new promoter that we developed in collaboration with Synpromics. The blue line is basically the line that you would see for our current therapy. 61 is this promoter that we're using in the blue line. And if you look now at our new promoter, you can see that we have a more than 10-fold increased expression of factor IX on a protein basis. So you know, the super, as you can see in the super 9 in the gray bars, the gray triangles. So this allows us actually not only to move this program forward, but it also, of course, has huge implications for other liver-directed gene therapies. Fabry, you know, this is lysosomal storage disease with storage of GB3 and lyso-GB3. Severe disease, ERT and enzyme replacement therapy, moderately effective and certainly not able to stop progression of renal disease, and particularly not effective in stabilizing cardiac disease. So there's a big unmet medical need here. And we started working on this, trying to basically target in particular these organs. So very briefly, we have a modified NAGA enzyme, which is basically mimicking now the efficacy of the alpha-galactosidase, which is the enzyme which is missing in Fabry. This is non-immunogenic because it is not alpha-galactosidase, which is basically immunogenic, as you all know, in Fabry. It's potentially more effective because we've shown that it's more stable in plasma. It's highly mannose 6-phosphate glycosylated, so it's taken up better than alpha-galactosidase, particularly in those end organs that I was just discussing, the kidney and the heart. We have preclinical proof of concept showing a really nice knockdown of GB3 and GB3 in Fabry mice. So this, again, we are bringing this to GLP tox stage now. The other one is SCA3. SCA3 is very similar to Huntington's. It's a polyglutamine disorder caused by CAG repeats in the ataxin-3 gene. Remember, Huntington also is caused by CAG repeats. The ataxin-3 protein acquires toxic properties because of this polyglutamine expansion. It leads to brain degeneration in cerebellum in the brainstem and spreads more widely in later stages. And it's characterized by ataxia and paralysis, and it will lead to death. It's actually not uncommon. It's quite common. It's by far the most common ataxia, and we think it's a fairly large market as well. Certainly, for example, larger than hemophilia B and maybe approaching the market of Huntington's. There's no medication right now, and there's very few competition in terms of silencing the SCA3 gene. And we are, you know, moving this program forward quite rapidly. As you can see here, we have 3 candidates, the MyCure A, B, and C, as you can see them there on the graph. We've done intracisternal administration of this product now in a SCA3 mouse model, and we showed really nice knockdown. This is, we predict more than sufficient to have therapeutic benefits. So this program really looks very good, and we're moving this rapidly forward in preclinical studies. So with that, I would like to summarize our progression and where we are with the pipeline, and save a little time for, you know, discussion. Thank you very much.
Thank you, Sandra. That's very helpful overview. So maybe I will just start by asking the question on hemophilia B program. I think the last update in February reported a longer follow-up of the factor IX level. Just wondering what measurement methodology was that? I think the reason I'm asking, because different company come up with different methodology, and then you have some inconsistency in terms of numbers.
Yeah. So all this was done with using a centralized one-stage clotting assay. And, like our competitors, and it's different for hemophilia A and hemophilia B. But, I mean, the general story, I think, is that if you look at chromogenic versus one-stage, there are differences. But they're on one line, basically. They correlate very well, but the absolute level is different. I think the one-stage clotting assay probably is what the FDA is going to ask for.
Okay. Yeah. Okay, and that was your feedback with FDA. Okay, that's very helpful.
But we measure both, and we also have central versus local. So, there's 3 different measurements actually there.
So, like in your hand, what is the correlation between chromogenesis versus a one-stage assay?
Well, we haven't talked about that, but I can tell you they correlate very well.
Okay. I think other experience depends on, like, it could be 60%, it could be 80-90%.
Well, I think the chromogenic under reports as compared to one-stage, and that's true for hem A and B. By the way, it's not something very special for gene therapy. I mean, it's been also reported for replacement proteins. So it's not something that we now suddenly find out. It's been there, and the question is, what is going to be used for regulatory end purposes. And by the way, in the history has it, for the development of other products, I mean, it generally has been one-stage. And one-stage, of course, is what centers will use in real life. That's what you use in real life.
Okay, very good. And also, for your phase 3 trial, you also do not have a pre-screening, right, for the existing neutralizing antibody. I'm just wondering if you can share a little bit more color on that, the rationale behind it.
Yeah. So the prevalence of anti-AAV5 antibodies in our hands, and we've now screened 350 donors from the US and Europe, males, females, young, old, what have you, and also some patients actually from South Africa. And the prevalence is not that different between we did 2 versus 5 versus 8. We screened all of them. So, but the avidity of those antibodies is much lower for AAV5. And we have shown in the clinic that up to a titer of 1 in 340, that was in our NT60 trial, we didn't see any interference with therapeutic benefit. Actually, that patient that had this high titer AAV5 neutralizing antibody was the highest expressor. And we know that this was a genuine AAV5 antibody because we know that that patient had been pre-exposed to AAV5. I mean, I won't go into details, but we discussed that at ASGCT. Now, in primates, we know that spontaneous levels all the way up to 1 in 1,000 don't interfere with therapeutic efficacy whatsoever at any dose of treatment. And probably the break point is even much higher. So in the population, you never encounter those high titers. So we are not worried about neutralizing antibodies, and we don't screen patients out because we predict we basically can treat 99% of patients without having any trouble there. Now the other point, of course, is T cells, because even if you screen out patients, for example, for AAV5 antibodies, for AAV8 antibodies, even those patients that screen completely negative, about 20 to 25% of them have a T cell response later. That's why, in those trials, everybody gets prophylactic steroids. And we've never seen a T cell response to AAV5. The AAV5 capsid is really different from AAV8, and we are in the process of characterizing both class 1 and class 2 epitopes, T cell epitopes, comparing the to 8 and comparing 5 and 8. So we'll report on further analysis of this, but from the antibody side of things, we know what's going on there, I think.
So another question is, the primary endpoint is the factor IX level. Based on your feedback, did the FDA provide any color on the threshold of factor IX level that would be approvable?
Yeah, so the answer is, as far as I know, the answer is negative. So So they didn't say it needs to be 50% or 55%. They said that you need to have a functional normalization of coagulation. And I think, if we speak to our KOLs and between 30 and 50%, that's what you do. I think, when discussing this with the FDA, far more important is going to be the longevity of the response and the predictability. Are you within that range? And do you see any decline? And so I think, we have answers to basically all those questions already, because the 61 transgene only is is almost the same as 60. And for 60, we have almost 3 years follow-up now, showing stable expression. And there's no decline whatsoever. And I don't expect that for 61. In terms of predictability, we predicted, this 30 to 50% range. All our patients are exactly smack in the middle of that. They all follow this curve that we predicted. So thus far, I think we're smack in the middle of all our predictions as far as that is concerned, and that's going to be far more important to the FDA than, do you have 50 or whatever.
Okay.
Yeah.
And that's very helpful. And then I'll just ask one quick question regarding the Huntington program. So what is your goal of a CSF reduction? And then also, what is your view? I think Roche now, comments on potential accelerated path based on the biomarker and early functional data together with natural history data. So what is, your thoughts on that?
Yeah, so we like that development, of course, because we do see a strong mutant huntingtin reduction as well. As far as mutant huntingtin, as a reliable predictor of response to therapy, I think we need to discuss just very briefly a few things. First of all, mutant huntingtin is made all over in the CNS, so it comes from the spinal cord and where have you. And actually, it's also generated in the rest of your body as well. Actually, the mutant huntingtin levels in your blood are significantly higher than they are in the CSF. So if you knock down mutant huntingtin by giving intrathecal administration of your antisense, it's unclear where that came from, that mutant huntingtin, because the sampling, of course, also is during the spinal puncture and the lumbar puncture, and so you don't know whether that mutant huntingtin came, for example, from the spinal cord. You could have silenced the spinal cord. So it doesn't predict whether the mutant huntingtin that we're talking about really came from the structures where Huntington's disease takes place. That's an important number one. When we see a reduction in mutant huntingtin, we know for sure it came from that place, because that's where we have the knockdown, right? So the other point I would like to again stress is that mutant huntingtin is only, a marker of many other things going on, and there's a lot of attention now on that small fragment, the exon 1 fragment. So to what extent mutant huntingtin levels relate to the far more toxic small protein that is transcribed out of the huntingtin gene, that's currently not known. So we're expanding our biomarkers in that direction as well. But I think it's a good, for us it's good, we like it, but I think you need to take into account that there are differences between these approaches, and that the mutant huntingtin as a biomarker between the approaches may not signal the same thing.
Okay. I know we are running out of time, and we'll save more questions at the breakout session that will be across the hall. Thank you very much, Sandra.